tell us a bit more about the DNA extraction step. What actually happens? You get the DNA or the saliva sample in the lab. What do you do to get the DNA out?
So DNA extraction is a very, normal thing that happens in labs all over the world. There's nothing special about how we do our DNA extraction, but the process is really trying to separate the DNA in the nucleus of the cell from the proteins and other components of the cell that otherwise would be in the way.
I see. And I mean, how much DNA do you get out of the saliva sample that someone sends in?
It's actually very variable. So depending on the person, we can get anywhere from 10 micrograms to over 100 micrograms of DNA.
Oh, wow. And does most of that variation happen just based on the number of cells that were in that spit sample or saliva sample?
We think so, but we actually don't have very good information on that. We know that some people's saliva is more watery than other people, and we know that, for example, infants do not often give us good data, and we think that's partially because of the wateriness of that saliva.
Well, for those people who might be wondering, what kinds of cells do you actually getting in my spit? What are the actual cells that you're extracting DNA from when you're looking at my saliva?
So in a sense, it doesn't matter in that almost all of your cells have your genome in there, and it's always the same genome. So we don't look at that too closely. We know that the major components there are epithelial cells, so the inside of your cheek, the skin cells there, and and white blood cells are the other major component.
Now, I do floss and brush my teeth, but I know that I'm sure I have some kind of bacteria or germs in there. Even still, is that a concern at all, worrying about sort of having other non-me DNA in my spit sample?
Again, it's not something we worry about too much. We know that people have a lot of bacteria in their mouths, and we know that from person to person there's huge variability in the composition of those bacteria. But for amplification, we're happy to amplify up all of your human and bacterial DNA and then let the sequencing or genotyping process sort out the human from the bacterium.
I see. So as long as it's human, it's good, but does that mean like I shouldn't go kiss anybody right before I give my DNA sample?
For the 23andMe test, we actually collect 2 milliliters of saliva from you, which doesn't sound like a lot, but it can take up to 5 to 10 minutes to fill that. So it's a lot of saliva. You would have to be kissing a lot to make a difference there.